Hello, everyone. My name is Marlia Knights, and I'm an undergraduate researcher in the RIO lab in the Department of Biology at West Virginia University. And my project is on the comparison of paratrophic matrix associated gene expression within tsetse species of varying vector competence. African trypanosomiasis, more commonly known as African sleeping sickness, has two stages of progression. Symptoms of the first stage include itchiness, fevers, enlarged liver or kidneys, and changes in sleeping habits. As the disease progresses into stage 2, symptoms worsen into personality change, confusion, seizures, and comas. If left untreated, death is likely. African trypanosomiasis is caused by parasites known as trypanosomes, which are transmitted to human hosts through an insect known as the tsetse fly. However, tsetse flies can differ in their abilities to sustain and transmit the parasite. This quality is known as vector competence. Therefore, it is important that we understand how vector competence is determined. This study attempts to answer the question, what makes one species more likely to transmit trypanosomes? So, we compared the expression of genes in the midgut of tsetse flies, specifically the paratrophic matrix, or PM, which is a structure made of chitin. The paratrophic matrix is pictured in the fly diagram in Figure 1. It acts as a protective barrier between the gut epithelium, or lining, and the blood meal, which is the sustenance of the tsetse fly. We examined two species of tsetse fly, Glossina brevipalpus, which shows low vector competence, and Glossina morsitans, which displays high vector competence. We hypothesized that G. morsitans, as it is more likely to sustain and transmit trypanosome infection, may show the downregulation of genes involved in the synthesis of the PM. The idea behind this is that the PM plays a role in the tsetse fly's immune defense. When a tsetse fly consumes the blood of an animal infected with a parasite, such as a trypanosome, the PM acts as a physical barrier between that infection, which exists in the blood meal, and the rest of the organism behind the gut epithelium. Therefore, it makes sense that a compromised PM will facilitate more trypanosome infections. In order to test this hypothesis, we follow the steps outlined in Figure 2. We dissected and homogenized G. morsitans and G. brevipalpus adults that had just emerged from their pupal states. We isolated the nucleic acid RNA from these samples using MasterPure RNA kits, as well as a DNase treatment to remove contaminating DNA. And we constructed a cDNA, or complementary DNA library, formed with reverse transcriptase. We then sequenced these libraries into apprehensible data with Illumina technology. Orthologous genes, or genes shared between two species due to common ancestry, were identified through these methods. These genes were also organized into COGs, or categories of orthologous groups, which detail what a specific gene's purpose is. Based on this data, we developed graphics called heat maps by a computer program known as RStudio. This allowed us to visualize how these genes are regulated in each species. By examining these heat maps, such as the heat map of information and processing genes in Figure 5, we were able to determine that both species of tsetse fly showed significantly more upregulation or green and yellow markings in the highlighted gut samples as opposed to the bacterium. Therefore, further analysis included gut expressions, not bacterium expressions. According to previous research, specifically an investigation into the composition of paratrophic matrix by the Liverpool School of Tropical Medicine, around 300 genes may contribute to PM composition. However, for the purposes of our experiment, only 123 of those are orthologous between G. morsitans and G. brevipalpus, with only a 1-to-1 expression. Of these 123 genes, 47 were differentially expressed, as shown in Figure 3. We were able to classify 117 of the 123 into known COGs, which are categories detailing the main function of a gene. After this process of organizing our genetic information, we carried out statistical tests including identifying genes with expressions of above a threshold of 430 in G. brevipalpus and 602 in G. morsitans, as well as identifying those genes that displayed significant differences in expression between species through calculating fold change scores and p-values. In 14 genes were differentially expressed, with expressions of above the threshold for either species. And of these 14 genes, 7 were found to have statistically significant differences in expression when p-values and fold change were calculated. Additional research by examining the genes through the vector-based search engine or through reading additional literature will give us insight into the biological significance of these genes. If we can understand the differences between tsetse flies, G. morsitans, and G. brevipalpus, this will also give us an understanding of the differences between high vector competence and low vector competence, and in what genes might promote or prevent the sustaining of trypanosomes within the tsetse fly. These results may be helpful in determining the new designs of new interventions towards vector control of tsetse flies and their transmission of African trypanosomiasis. Thank you so much for your time and have a great day.